All right, so this should be recording. So here's Carb Lecture 3 and some of the things we're going to go through. We're going to start looking at the energy pathways. So we'll talk about glycolysis and go through that. But mostly we're going to talk about the aspects of it that are regulated. We'll talk about why that's important, both in a cell and the larger kind of metabolic systems throughout the body. And then we'll look at galactose and fructose metabolism. We'll see if fructose deserves the bad press that it gets, right? We're not going to cover hexose monophosphate shunt or ethanol metabolism in here. So the parts of the textbook that talk about those, don't worry about those. We'll talk a little bit about hexose monophosphate shunt in 329 when we talk about vitamins like thiamine, so that's coming. I've showed you this before, right? And we said, well, if you were going to explain to somebody how we get most of our energy, if they said, well, how do we get energy from food? And we said in one or two sentences, what would that be? Yeah, so their structures basically allow them to enter pathways that generate energy mostly by the oxidation of the molecules that provides electrons for ETC. Right, so it's mostly through ETC. Krebs cycle doesn't generate a lot of energy on its own, but it gets electrons, and the electrons go to ETC. So carbohydrate, fats, and proteins can support Krebs cycle and ETC. Now, the most significant energy sources are carbohydrate and fats. We don't get much energy from protein. It's usually somewhere between like 2 to 5%, something like that. Okay? But what we're going to talk about is glycolysis now, but you can see glycolysis is really important to regulate because it provides not only Krebs cycle energy, but it provides our ETC, but we can get energy through glycolysis. And this is a little bit of a combined slide from before, but this is tissue-specific energy pathways, so a little bit of review. If we look at the red blood cell, remember their main function is transport oxygen, so they just run glycolysis. If we look at the brain, here's the outer membrane of the brain cells, for example. These inner circles are mitochondria, obviously. So the brain cells will typically run on glucose. They'll run glycolysis and then Krebs cycle. So does aerobic and anaerobic metabolism occur at the same time? Yes. Does aerobic and anaerobic metabolism occur at the same time? It can, yeah. I mean, so if you look here, for example, I mean, glycolysis runs through here. That's anaerobic, and yet it's supporting its Krebs cycle and ETC. So does ETC occur in brain cells? Absolutely. An indication of that is, think about how important oxygen is to those tissues. But it does occur at the same time. Throughout the day, it's kind of a percentage of one or the other. Like right now, you're doing a combination of aerobic and anaerobic metabolism. At this point, you're getting most of your energy from aerobic metabolism, but it's a balance. It's rarely one completely or the other. Okay? So if we look down here at the liver, oh, and here's ketones. Remember, ketones can support energy metabolism in the brain during starvation. Okay? And we'll talk a little bit more. We'll talk a lot more about ketones. We talk about lipid metabolism. I'll show you how they're made and all that. Here's the liver. Wow, a lot more going on in the liver, right? These are liver cells, for example. They have mitochondria, but they'll run glycolysis, Krebs cycle, ETC. They can also store glycogen. They can take in glucose and export it. It's the only cell up here that'll do that, remember? And again, why are these bidirectional? Because they'll do gluconeogenesis that we'll look at next time. And here's another thing. We talked about this before. Oh, you can cross this out. Pentose phosphate pathway. It's also known as hexose monophosphate shunt. Remember that fun stuff probably from biochem? No? Okay. And glucuronides, those are breakdown products, so don't worry about that. You can cross that out too. So over the whole quarter, at least you can cross off 2 things, right? Okay, you can get rid of those. Now look what else it's doing. This is a fat cell making. What's palmitate? What's palmitic acid? That's a fatty acid. So this, in this case, the liver cell taking citrate and converting those carbons to palmitate. So when do you think this happens? If this liver cell has a lot of energy already, it can do this. So this is, for example, overfeeding. Liver cells have maximized ATP amounts, and they'll start doing this. We'll look at how that happens coming up. And here's a muscle cell. Right, glucose goes in, glycolysis, they have mitochondria, so we're running Krebs cycle, ETC here. Here's lactate under anaerobic conditions. It's pushing that out. Oh, here's lactate coming into the liver. Lactate only goes out of the muscle cells in general. And then here's glycogen. It goes back and forth, but doesn't come out. And I'll give you more details on that coming up. So again, the liver's really important in glucose metabolism because of some of the things it can do. Before we start glycolysis, let's talk about some of the expectations as we go through this. As we go through these pathways, you're going to be able to briefly define the pathway, beginning and end product. So you'd say glycolysis is glucose to pyruvate, and then I'll tell you which structures you need to know. And then we'll talk about regulation of it and the significance of it under certain conditions, like what happens during fasting, after a meal. You'll know the intracellular locations, tissue specificity, and key enzymes and how they're regulated. Oops. So here's a way of looking at glycolysis. Okay, so in your notes somewhere, say, use this diagram for glycolysis, not the textbook. This is from an older edition that's right. Most of it's right. The 6th edition of the textbook has a mistake. So use this one. Their pathway's wrong. Okay? You can cross out hexose monophosphate shunt, also known as pentose phosphate. Also do this. So you're going to add into this where it goes glucose 6, and then you already have glucose 1. So from glucose 1, you can go to glycogen, because we're going to talk about that process. So for example, you can look at this diagram, just go, oh, okay, based on this, can we use, oh wow, that's not very good. That says galactose up there. Galactose up there converts to galactose 1-phosphate. What tissue would that happen in? Liver. Remember, the liver takes up 100% of the dietary galactose. So if you look at this, you can say, wow, if I consume galactose in my diet, can it support hepatic glycogen? Yeah, because it can convert to glucose 1 and then glycogen. Okay? So galactose is kind of neat because of that. Now, if we go through this process, we have glucose going in, glucose 6, fructose 6-phosphate. Remember all these? This is glycolysis. Here's glycolysis. Goes like this, then wraps around like this. So it's not linear on this diagram. And I like that because it shows where fructose comes in and galactose, and it kind of separates it into energy investment phase, energy production phase. Remember that? So it would be fun to memorize all the names and structures on here. You don't have to do that for this class. And when I give you a study guide for this, I'll list out the structures that you have to know. You don't have to know all of them. But if we go through here, we can see we have, so here's glycolysis, glucose to glucose 6. That's the first step. Takes an ATP, and the phosphate's added on that hydroxyl group of carbon number 6, so we call that glucose 6-phosphate. That converts to fructose 6-phosphate. And you can see, for example, this reaction, the reaction number 2 up there, is bidirectional. This is unidirectional. And this is done by which enzyme? Glucokinase or hexokinase? Right? Depends on the tissue. And now we have an enzyme, now we have this reaction, and now we have another unidirectional reaction. A lot of times in these, unidirectional reaction involves ATP. That's why it runs one way. And now we invest our second phosphate here from ATP, so at this point we've used 2 ATP. And we're at fructose 1,6-bisphosphate. And this structure's wrong. What's wrong with this structure? This should go through. So we need a hydroxyl group here. And this is wrong. There shouldn't be a hydroxyl group here. Got that? This should extend. So we should have an OH here. And we don't need it there. So here's fructose 1,6-bisphosphate. Okay. Now, so 6 carbons here, and now look what happens. Remember, this is where it splits. So fructose 1,6-bisphosphate splits into 2 different molecules. Each of those molecules has a phosphate. One is dihydroxyacetone phosphate, DHAP. And the other one is glyceraldehyde 3-phosphate. Both of these come from this molecule. Each of them has 3 carbons. And now, G3P becomes 1,3-bisphosphoglycerate. So it's modified, and we'll talk about this step in a second. And then, in this case, this phosphate, see that little squiggly line? That means it's a high-energy phosphate bond. So this phosphate can be added to ADP to get ATP. So after all that, we get an ATP out of it. So we invest 2, we got 1 back. This is modified to become, then 3-phospho becomes 2-phosphoglycerate. Basically, the phosphate moves from here to here. No energy details there. 2-phosphoglycerate becomes phosphoenol pyruvate. I'll show you that again in a few seconds. Actually, you should probably spell that one. phosphoryl pyruvate. That becomes pyruvate. And when it does, it generates another ATP. So we invested 2, and we get 2 back. So we're at 0. How many net ATP do we get from glycolysis?
2.
Plus 2. And the reason we do is because we repeat these steps. Remember that? So in other words, G3P becomes 1,3-bisphosphoglycerate, and then DHAP becomes G3P. So they both go through this process. So all 6 carbons go through it. That's why you end up with 2 pyruvates. Remember all that? That seems like a lot to get 2 ATP. What's the advantage? Don't need oxygen. So anaerobic energy production is expensive. So now imagine if I'm doing high intensity exercise and I'm doing all of that just to get 2 ATP, I need a lot of glucose to maintain a significant effort over a period of time. So it's one of the reasons carbohydrate can become limiting during certain types of exercise. We don't store a lot of it, so we can deplete it relatively quickly. The harder and faster we're going, the faster it goes away. And once that glucose is gone, fat's the only is the only substrate left, really, and that requires oxygen. Sometimes that's what makes a person slow down, because they need more oxygen.
What kind of physical activity translates like 2 ATP? Like what can you do with 2 ATP?
Oh, nothing.
So
It takes a ton of, it takes a lot of ATP and a lot of glucose molecules. I wouldn't be able to tell you how many numbers of glucose molecules it would take to, run from here to our building. I don't think I ever knew how to calculate that. I probably could, but not off the top of my head. Yeah, so it's very expensive. Now, and then we'll also talk about how fructose plugs in and how galactose plugs into that, because can they enter glycolysis? Yep. Okay. But they enter at different points. Right? Sometimes you hear people say, oh, fructose is harmful. Have you ever heard that? Stay away from fructose. It's harmful. Depends. It does enter into glycolysis. Galactose does too. What's different about them? Where they enter. Here's one of the most important regulatory points of glycolysis right here. And we're going to talk about that because cells can regulate this step. So when galactose goes into glycolysis, it's regulated. When fructose does, it's not. See the way, well, this should say major and this is minor. We'll talk about that. Most fructose goes this way. It goes into glycolysis after the regulated step. So that's why too much of it can be problematic. And we'll talk about all that.
where did this to glycogen fit into the diagram?
We'll talk about glycogenolysis and gluconeogenesis and everything, and basically how they're related. But and one of the reasons is because when glucose goes into the cell, it has an option. It can either go to glycogen or it can go through glycolysis. So I would just wish that diagram had glycogen on there. That's all we did.
So would it fit in after the glucose 1? It would just be like another arrow.
Yep. It goes this way. So there's the glucose 6, then it goes glucose 1 phosphate, and then glycogen. So it would be off of there. Yes?
Does DHAP wait for the G3P to finish or to like make it to pyruvate before it goes into the cycle?
Oh, good question. No, those are in equilibrium. So as soon as this one leaves, this one shifts over. So it kind of runs that way.
The steps that are reversible, would it ever reverse mid-glycolysis?
I suppose it could.
Why would they do that?
Why would they? Well, in general, the way this is regulated is if this is glycolysis going this way, generally when glycolysis is elevated, gluconeogenesis is downregulated. So most of the pathways in the cell, it's either running this way or that way. Yep. And there's a few different ways it's regulated. Here's the ATP production. The reason we're going through this is you guys are going to be able to add up all the ATP that comes from this. Okay? So we went through this. We said here's the ATP where it's generated. And then this is an interesting step here. And this step 6 is related to this right here. See, it says net 2 ATP. But this says by substrate level phosphorylation. What does that mean? Yeah, that's a direct phosphorylation of ATP. So in other words, that's here. This ADP is phosphorylated to ATP. This ADP is phosphorylated to ATP. That is a direct energy production step. This one generates energy indirectly. Okay? That's where this comes in. Now this step is, this process is called the malate aspartate shuttle. And it involves step 6 of this glycolysis process. So a couple things that we've said already is that glycolysis occurs, what part of the cell? Cytoplasm, cytosol. How do we get most of the energy in a cell, typically? ETC, Krebs cycle, and where are they? In the mitochondria. Okay? And if you remember from previous classes, or a couple slides ago, we said, well, ETC gets those electrons from things like Krebs cycle, but the ETC can also actually get electrons from step 6 of glycolysis. Have you talked about this before? Okay. So in this process, what's happening is, here's the cytosol, and here's NADH in the cytosol. And this NADH is going to convert oxaloacetate to malate. So this says cytosol, but actually, you know, you could look at it like, here's the mitochondrial inner membrane. So you're going to put in your notes, mitochondrial inner membrane. Well, what did we say about the inner membrane? Porous, non-porous? Non-porous. Why? ETC's there, proton gradient. It's very stringent to the passage of other molecules. So NADH does not go through that membrane. Okay? So here's what happens. Malate can go through that membrane. So I'll simplify this, and you can draw yours like this. So here's malate. We're in the cytosol. Here's the matrix. Here's the mitomatrix. Malate can go through. There's a shuttle for it. In other words, there's a transporter. Where does the malate come from? Oxaloacetate. Where have you heard those molecules before? Krebs cycle. So relatively common molecules. They convert back and forth. In the cytosol, this can happen if NADH is present. Where can we get this? We'll say step 6. Oops. Step 6 of glycolysis. Can provide that. NADH during this process converts to NAD. We write it like this. Remember formal charges, we'll talk about that in a minute. So what's happening to oxaloacetate in this case? Is it being reduced or oxidized? Which of these two molecules has more electrons? NADH. What's happening to the electrons? Adding to oxaloacetate. So oxaloacetate's being reduced. Here's maybe an easier way to do this. This has two electrons that are passed on to malate. Now malate can go across. NADH can't go across, malate can, so the electrons are basically handed off to malate. Malate goes in, and now look what happens. On the inside of the mitochondria, we basically reverse that. Now we go malate to oxaloacetate. Now where are the electrons? Now they're here. And now oxaloacetate can convert to aspartic acid. What kind of molecule is that? That's an amino acid. So nitrogen's added. Convert oxaloacetate to aspartic acid. Why do you do that? Because aspartic acid has transporter. Oxaloacetate doesn't. What's the point of this? Energy production. This goes to ETC. Because that's where it is in the matrix. What kind of energy can you get from this? ATP, how many? Generally we say 3. And we'll go through ETC coming up. Anytime NADH contributes electrons to ETC, we get approximately 3 ATP. When NADH contributes electrons to ETC, it generates 3 ATP. So, how many of these would you generate from one glucose molecule? 2, because you do these steps, this step, twice. So you get NADH 2 times. So during anaerobic conditions, you get net 2 ATP from glycolysis during anaerobic conditions. What about aerobic conditions? You get how many more? You get 6 more. So essentially you get 8. So during aerobic conditions, the malate aspartate shuttle can add 6 ATP. That's because Krebs cycle and ETC are functioning. And we'll add all those up coming up. And one of the things I'm driving at is, did you guys talk about this in other classes? This is a good number to remember. What's the maximum number of ATP that can be produced from the metabolism of one glucose molecule? It's the maximum number of ATP. Have you ever heard that? Remember what it is? 30 something. A lot of times you'll hear, oh, it's 36. Sometimes you'll hear, oh, it's 38. Why is there ambiguity? It can, kind of. Okay, so this is the malayed aspartate shuttle. It uses NADH. Liver cells use this one. Muscle cells use a different one that's based on glyceraldehyde. And it uses, and you don't have to remember that kind of stuff, it uses FADH2 instead. So imagine this has replaced FADH2. FADH2 here. How many ATP do you get per FADH2? 2. You do that twice, you get 4 instead of 6. That's why it's 36 instead of 38. So it depends on the tissue. But if I ask you what the maximum number is, you're going to say 38. So far we have 8 of them. So when we go through Krebs cycle and ETC, we'll come up with another 30. These are going to be quick. Memorize that. Memorize that. Just kidding. This, have you ever looked at the structure of NADH? Give you extra credit right now. Can anyone write out what NADH stands for? Spelling correct? What is it? Should we do it as a class? Nicotinamide adenine dinucleotide. Right? Yeah. Where's it from? From our diet. What's it derived from? What do you think? What does that sound like? Nicotinamide. Any vitamins? Ah, niacin. Ah. There's 2 forms of niacin, and we'll get into this next quarter, I hope. Nicotinamide, nicotinic acid, they're both forms of niacin. Niacin is what kind of vitamin? What category? It's a B vitamin. Have you ever heard that? B complex. Got to have B complex for energy production. If you're tired, is anyone in here ever tired? Do you have issues concentrating for long periods of time? Sometimes hard for you to focus? You could need a B complex vitamin supplement. That's what they'll tell you. They'll say, oh, because you got to have it. It's important for energy production. And people go, oh, I'll do that. I'm still tired, but I'm out $30. The reason you can say that is because the B vitamins, including niacin, are required for energy production. That's how we make NAD. Truth of the matter is, unless you're deficient, additional amounts aren't going to increase your energy output. But it is derived from that. And here's why it's important. The top part of this is nicotinamide. So there's the B vitamin part of it. And then we have nucleotides that are added in the form of phosphates. So we call it nicotinamide. Adenine is over there, that structure. So it's this big structure that can bind electrons. Because of its size, it would need a transporter to get into the matrix. There's no transporter. So what our cell does is it makes some NAD for the matrix, it's going to make some for the cytosol. Because there's no transporter, it can't go through. So the cell uses these shuttles. That's the point of this. NAD does not go through that membrane. So what's the point of the malayed aspartate shuttle? Here's what it is. The purpose of the malayed aspartate shuttle is to maximize ATP production by shuttling glycolysis electrons into the matrix for ETC. So when does that happen? Aerobic conditions or anaerobic conditions? Aerobic. It's got to be aerobic. Otherwise, you only get 2 ATP because ETC is not functioning, and this won't happen. And just as kind of a side note, remember this? Everyone got that definition? As kind of a side note, let's say this is anaerobic conditions. This NADH, what tends to happen to it? What's it used for? this is going to happen. It's going to go NAD to NADH. Now you have that NADH. Under anaerobic conditions, what is it used for?
Storage of electrons. Is it for fermentation of the lactic acid?
Conversion of pyruvate to lactate. That's what pushes that forward. And then what happens? Then you get NAD, and it's used here. So under anaerobic conditions, it cycles. Under aerobic conditions, it's pulled out because you're not making lactate. So before too long, I'm going to ask you how high-intensity exercise generates lactate. And that's another thing that this is going to plug into. Got all that? And we all know lactate's what makes our muscles hurt when we exercise it. No, it doesn't. It increases the potential for pain during that type of exercise, but it's not the lactate that's actually doing that. Let's not go into this unless you want to. This is just showing that this is where on that molecule we are binding the electrons. So one of the important points of this is these up here. What is that? That's hydrogen. What's this? That's a proton. What's the difference? The electron. So this is why we write this as NADH plus a proton. Because when you add the electrons to this, you end up with NADH. One of the electrons stays with hydrogen, the other electron's added to this ring. So the electron is pulled off of the hydrogen, that makes a proton. That's why you always see it like that. You ever wonder why? That's why. So that kind of cleans up the math in that case. That's where that proton comes from. So my point is, a hydrogen is different than a proton. So if I ask you to explain how ETC works, if you need to say proton gradient, don't show me a hydrogen. Don't say hydrogen. That's different than a proton. Okay, so putting that in your head right now, that's a difference. And here's FADH2. It's simpler. It actually binds two hydrogens instead of one hydrogen, one electron. That's why we write that as FADH2. How can we make FADH2? Look at the name. You all knew that. FAD is flavin, adenine dinucleotide. Where do we get that from? Riboflavin, another B vitamin. Okay. So let's talk about regulation of metabolic pathways. And a lot of this is going to be review, but I think it's worth it. So this is a point where you go back to chapter 1 or read the last couple pages in chapter 1, where before I said skip it. So you can read about catalytic proteins and enzymes, and it'll say catalytic proteins and all that. Here's the key points. And a lot of people don't realize this, but most enzymatic reactions are reversible. Why? Because the same enzyme can run it either way. Remember that? So for example, if we have this pathway going from A to E, and we want to regulate this pathway, let's say we want to increase the amount of E that's made from this. Let's say, for example, we want to go from glucose to pyruvate. It's regulated at the unidirectional points, not the bidirectional points. So we wouldn't increase the activity or the levels of this enzyme, because if we have more of that, we're not pushing flux one direction. It can go either way. So we would regulate this enzyme. And if you look at the glycolysis pathway, you can see those unidirectional arrows. Those are important because they tend to be points of regulation. There's a few different ways to regulate it. So we're going to review those. If, for example, we have an enzyme, obviously a protein, it has a gene that can be regulated. So we can regulate transcription. Once we have mRNA, that mRNA can be either retranslated or degraded. And this is called mRNA decay or mRNA half-life. Have you talked about that in other classes? Really important point of regulation. Nobel Prize kind of level of importance. But then if we do translate that, we have an enzyme. We can still regulate that process or that enzyme's activity. And in some ways, this is a lot faster way to do it. Because imagine in this case, we have to go from a gene through transcription, through translation, and now we affect the enzyme's activity. A faster way to do it is just take an enzyme that's already there, turn it on or off. You don't have to wait for transcription and translation. So sometimes what happens in this case, this is called enzyme induction, going from the gene. We induce expression of the gene. Some hormones work like that. That's considered slower than the other mechanisms. There are some enzymes that are regulated allosterically. Remember that? Those enzymes have an allosteric site. And I'll show you diagrams of this. In that case, a modulator can bind to that site and affect the enzyme structure and activity. Not all enzymes are like that. Some enzymes are covalently modified. And for years, the most famous covalent modification is phosphorylation. Add a phosphate or take it off. Probably another type of modification that is kind of a new area of research is acetylation, whether or not acetyl groups are added. And they're finding that that's really important for aging and lots of other processes, but we're going to talk about phosphorylation. So here's allosteric regulation. In this case, we have the binding of a modulator, and that affects the enzyme's activity by altering the shape of the enzyme and therefore its active site. So this is a space-filling model of this. Here's the enzyme let's say before the modulator binds. Here's its structure after. So these allosteric modulators, even though they are binding non-covalently, they tend to change the structure of the enzyme, and that's what affects its activity. So this is actually a transaminase, an enzyme that's involved in amino acid metabolism that's either turned on or off by a modulator. This diagram is kind of showing that. Let's say this is the enzyme structure in a less active form. The C is for the catalytic subunit. Here's a regulatory subunit. In other words, active site, allosteric site. Active site, allosteric site. You can see they're different locations. But the allosteric site can change the structure of the enzyme and affect the active site. So the M is for modulator, and in this case it's showing a positive modulator. But some modulators are negative. So we'll go through that example. You can see it's bound. Structure of the enzyme is different. Catalytic site or active site is different. Now substrate can bind and be modified. So here's another important point of this. The modulators bind non-covalently. Modulators bind non-covalently. To the allosteric site. Which means the modulators can kind of come and go, combine and then be released. Here's another important point. Some allosteric sites bind either a positive or negative modulators. Same site. Some allosteric sites can bind positive or negative modulators. Now that's interesting, because now you got the same site that is now responding to different scenarios in a cell. If something goes up or down, that enzyme can modify its activity either up or down, depending on who's binding. Again, remember the context of this is, how is glycolysis regulated? So we're going to apply this to that. Okay, so keep that in mind. Here's another major mechanism of regulation. This is phosphorylation. This is review, I'm sure. This is when phosphates are covalently added. And they're added to the side chains of serine, threonine, or tyrosine because their side chains have hydroxyl groups. Right? These are amino acids. So in this case, a phosphate group is added to the side chain of an amino acid. And as this diagram implies, here is the enzyme before phosphorylation. Here's a hydroxyl group. Here's serine 14. That means this enzyme actually is a dimer, has two subunits that are the same. So it's got serine at its 14th amino acid. And this hydroxyl group is covalently modified to have a phosphate now. And you can see that phosphate now has changed the structure of the enzyme. And in this case, the enzyme becomes more active. Now the active site is more readily available for substrate. So it's a structural change. And now what happens? In order to do that, it takes an enzyme because it's covalent. And we're going to start having more and more fun with naming enzymes. The enzymes that add that phosphate group are kinase. It's a kinase. And a lot of times the hints for an enzyme's name have to do with what it does and its substrate. So the name of this enzyme is phosphorylase kinase because this enzyme is phosphorylase. So phosphorylase is its substrate. And I'll give you more examples as we go through that. Now, kinases add, what kind of enzymes remove the phosphates? Phosphatase. So kinase adds the phosphates, phosphatase removes them. Different enzyme. This is where it gets a little bit confusing. So this enzyme's name is phosphorylase phosphatase because it's removing phosphates from phosphorylase. Got that? So now think about what's happening here. In order for this process to go forward or backwards, we regulate these enzymes. And that's what causes the effects. So in some ways, it's a little bit clunkier than the allosteric, because now you got two enzymes involved. But it's a very effective, common way of regulating enzymes. So we're going to look at some enzymes that actually are regulated by both allosteric and phosphorylation. Okay? But again, here's the point. The enzyme's already there. We can turn on or off. So that's what that means. So hormones often use this mechanism to regulate an enzyme's activity. Again, either up or down. Now let's kind of start applying these to glycolysis. We're going to talk about the two major points of regulation for glycolysis. And that's step 1, which is glucose to glucose 6. And step 3, which is fructose 6 to fructose 1,6 bisphosphate. We've already talked about hexokinase and glucokinase. So it's a major point of regulation. Again, it's a unidirectional reaction. Hexokinase, glucokinase run this forward. Takes a different enzyme to run backwards. That first step is regulated by a few different things, and one is which enzyme isoform is it? Is it hexokinase or glucokinase? So we went through some of this before. We said hexokinase is there in muscle and adipose tissue, extrahepatic tissue. It's negatively regulated by glucose 6-phosphate, and it has a lower Km than glucokinase. I'll show you the curves in 1 second. Glucokinase expressed in the liver and beta cells. It's not negatively regulated by feedback. In other words, it'll keep phosphorylating glucose and has a higher Km than hexokinase. So let's look at these curves. Have you seen these before? Okay, so these are basically Km curves. Now this is why this is important. Like sometimes you'll do this in biochemistry or whatever, and you get all these enzyme kinetics, and you go, yeah, I don't care about that fungi's enzymes. For us, these enzymes are really important because Glucokinase is on liver cells. Its Km is really high. Its Km is 10 millimolar glucose. And the reason we can say that's high is look at its Km compared to hexokinase. I know it's 6 o'clock at night, it might have been a long day, but what's the difference there?
How tired?
No, I mean, like the number-wise, what's the difference there? 100-fold difference. It's 100 times difference between glucokinase and hexokinase. There's a huge difference in their KMs. We say a high KM means a high capacity or fast enzyme. Look how fast glucokinase is compared to hexokinase. It's kind of 100 times faster. And the reason that's important is glucokinase is on liver cells. Liver cells are exposed to significant increases in blood glucose. Glucokinase is fine under those conditions. Okay? So again, let's take the numbers and kind of apply it to real life, because that's an advantage we have in nutrition. So under fasting conditions, normal fasting blood sugar or glucose is here. Let's say 4 millimolar, which is in that 70 to 110 range if we convert it from millimolar to mg per deciliter. So there's fasting. Now, if you look at the intersect of this and we say, oh, here's the Km curve for glucokinase, and we say, oh, well, here's where they meet. So in other words, under fasting conditions, how fast is glucokinase working relative to its maximum rate? Another way to say its maximum rate is its velocity, or Vmax. So this is the Vmax. So if we say, oh, here's 25, 50% of VO2 max, there's 75 and 100. So now if we look at this intersect, we can say, oh, under fasting conditions, glucokinase is only working at approximately 30% as fast as it can. Okay? Now, when we go home and eat tonight, if we have a lot of carbohydrate and the blood glucose goes up significantly, it's pretty reasonable that concentration is going to double or potentially triple. Right? This is the portal vein. So blood glucose can go up a lot. Let's say it goes up 2.5 times to 10. Now we look at this intersect. We say, oh, after a meal, how fast is glucokinase working relative to its max? Half. Even after a meal, glucokinase is nowhere near working as fast as it can. In other words, the more glucose that goes into that liver cell, the more glucose can be phosphorylated. So the liver's really good at trapping glucose. Now, because it's 50%, we say, oh, that's half Vmax. And remember I said this before, what Km is? Km equals substrate concentration at 1/2 Vmax. And when you do these in the lab, essentially when you set these up, you can calculate it by increasing glucose, but leave your enzyme concentration the same. You can do these curves. And remember, it kind of falls off like this. And eventually you say, oh, it gets out there, there's my Vmax. And now you take 50%, and you say, oh, here's 50%. We know it's 10 millimolar. That's why that is. So you just go, boop, drop down, 10 millimolar. That's why we know that. So here's where it's interesting. Hexokinase is pretty much everywhere else. Let's focus on muscle. Let's say, for example, fasting conditions, blood glucose is here. Under fasting conditions, in muscle cells, how fast is hexokinase working relative to its maximum velocity? Like 90%. Under fasting condition, hexokinase is almost saturated. So when you increase blood glucose, like after that meal, first of all, the muscle is not going to be exposed to as much glucose as the liver because it's not exposed to the portal vein. But let's say it does double. let's say it goes up to 8 or something. Hexokinase is basically saturated. So it's not a very fast enzyme. So after a meal, the liver has a big advantage for trapping glucose. Is that advantageous? Compared to muscle, it is. Why? Because the liver will turn around and put that glucose back into the blood later. Will muscle do that? Nope, it won't. So now think about why that's set up. What do we have more of? Muscle tissue or liver tissue? We have a lot more muscle tissue. So it would be a problem if all that glucose could go into muscle and be trapped at significant amounts, because then other tissues wouldn't have access to it. So it's set up that way for a reason. It kind of protects from oversaturating the muscle. Now, one more point of this is if you reverse that and say, well, during fasting conditions, which cells are better at trapping glucose? The muscle. Because that has a low Km, it has a high affinity. That low Km means at low concentrations, it works efficiently. I think that's the end of, so we'll end right on that kinetic curve. Is that good? Okay, so I'll stop recording this and I'll send you an email as soon as I post this. If you have any questions, let me know. If you guys want to meet later on in the week, let me know.
Are you gonna have office hours on Tuesday? Since we have no-
Oh, that's probably a good idea, huh? 1 second.